Hello, everyone. Welcome to today's webinar. I think we're ready to get started. I'm Dan Kraus. I'm a senior scientist here at HEI. Before we get started, I'm just going to go over a few little housekeeping announcements. So just so you know, all attendees are muted for the duration of the webinar. We have 4 speakers today, and all questions are going to be reserved for the Q&A at the end. So during the sessions, please use the Q&A function to add in any questions you have. You can also upvote questions that are there that have been posed by others. If you have any logistical difficulties or questions related to the video or to where the video will be available afterwards, use the chat box. Just so you know, the slides and the video will be available in the coming weeks, so just check our website for details. And I really do encourage you to use the post-webinar survey, which will give us information about what worked and what didn't work, and what you liked and what you didn't like. And on our website, you can see the registration information for the upcoming sessions. So today's session is going to be about the COVID-19 pandemic, air pollution, and health. We have 2 session chairs. We have Barbara Hoffman from the University of Dusseldorf and Jeff Brook from the University of Toronto. Both Barbara and Jeff are members of HEI's research committee. And at this point, I'm going to hand off to Barbara, who will introduce the speakers in the session.
Well, thank you, Dan, for the introduction, and welcome to everybody to this webinar today. Just a very short introduction into the topic we are touching today, and not that I want to get you into a bad mood, but after the pandemic is before the pandemic. Now that we're seeing a little bit of light at the end of the tunnel, it is very much time to think ahead and look at where we will be going and what's ahead of us. So we know that over the past 30 years, there's been an increase in pandemics worldwide, and this increase is actually bound to continue. About 75% of infectious disease outbreaks originated in animals, the so-called zoonoses. And this is the reason for that lies very much in our lifestyles. We are erasing the buffer between wildlife and civilization by, for example, by deforestation, by destruction of ecosystems leading to loss of biodiversity, by wildlife trade and factory farming. And all of these practices increase the likelihood of a jump of pathogens across species and into humans. And like John Oliver, a famous comedian, said, it's like we are active with our lifestyles, we are actively trying to start a pandemic the way that we are behaving right now. So there's obviously a need for fundamental societal changes. We must not go back to business as usual after this pandemic. One of the approaches here and one of the rethinking that we need to do is the so-called One Health approach that really looks at the health of ecosystems of the world and treats our Earth as one large ecosystem. So what does all of this have to do with air pollution? Well, very early in the pandemic, there's been quite a few reports about the role of air pollution in spreading the disease, in enhancing susceptibility to infection, and inducing more severe disease. And we will talk about this and some of the biological underpinnings. Mary Prunicki will present the link between air pollution, COVID, and disease. Afterwards, we will take a closer look at racial disparities in COVID-19 and air pollution as one contributing factor to these racial disparities. This will be done by Tess Mersha from Cincinnati Children's Hospital. After that, Paul Villeneuve will highlight the methodological challenges and also the related public health messaging around air pollution and COVID-19. And finally, we will take a look at one example for a One Health approach, looking at health of animals, animal populations, and human populations, which will be presented by Lidwin Smit from Utrecht University. This is all followed by a session of questions and answers, which will be moderated by Jeff Brook, my co-chair for the session. Now here are our 4 speakers for today, and we will start with Mary Prunicki. She is a senior scientist and director of air pollution and health research at the Sean Parker Center for Allergy and Asthma at Stanford University. She received her PhD from Northwestern University and her MD from Southern Illinois School of Medicine. Dr. Prunicki investigates the impact of environmental exposures on health, especially the immune system. She researches the impact of air pollution and wildlife smoke in all age ranges. Most recently, she has begun to investigate the intersection of air pollution and COVID-19, focusing on the similarity of the immune dysfunction caused by air pollution and COVID-induced cytokine storms. And with this, Mary, I'd like to hand over to you to present your talk. Mary, we cannot hear you yet. You might still be muted.
Okay, can you hear me now?
Now it's fine. Now we can hear you. Okay, go ahead. Floor is yours.
All right. Well, good morning, everyone. So this morning, I'm going to be giving a general overview of the association of air pollution and COVID with health, but then I'm really going to focus on the why, at a biological level, this association makes sense. Let me see. Okay, so the what, when we talk about air pollution versus COVID, as we know, both short and long-term exposure to air pollution, especially studies involving PM 2.5 and nitrogen dioxide, indicate that the exposure to the pollution may contribute to increased COVID-19, both susceptibility, morbidity, and mortality. Both air pollution and COVID have disease similarities. So we know from air pollution studies that, there's been a large body of evidence showing that air pollution impacts many organ systems. We know that, for example, from the wildfire research, when someone's exposed to wildfire, there's an increase in both respiratory and cardiac outcomes. We know from air pollution studies that the pollutants can impact the brain. It can increase dementia, it can increase stroke. There's also associations between obesity and air pollution levels, high blood pressure, diabetes, and the list goes on and on. When we talk about COVID, we know that certain chronic diseases are associated with increased risk of COVID hospitalization. So, for example, if someone has asthma, they're going to be more likely to be hospitalized with COVID. High blood pressure, obesity, diabetes, when people have renal disease, and if they have a combination of chronic conditions that increases their susceptibility to be hospitalized with COVID and have a more severe outcome. So in general, when we talk about air pollution and COVID, both of these exposures cause widespread inflammatory changes within the body. And we can measure these impacts by looking at different types of biomarkers in the blood. And for each type of whether it's air pollution or COVID, we can see that there are similarities in the elevation of inflammatory markers and immune dysfunction, which I'll be focusing on more later with that. When we look at PM 2.5 exposure as an example of air pollution, most of the research, the majority, when we focus on the health impacts, have looked at PM 2.5 levels. And worldwide, you can see that other parts of the world fare much worse on their exposure to PM 2.5. When we plot life expectancy as a function of the amount of exposure to PM 2.5, we can see that across the globe, exposure to PM 2.5 is related to, is shortening life expectancy. Here in the United States and Canada, our lives are cut short by more than 4 months on average, just from exposure to PM 2.5. But if you go to other parts of the world, the rates go up to almost a 2-year decrease in life expectancy simply from exposure to PM 2.5. When we talk about the association between air pollution and COVID-19, there's been several studies that have come out that have shown a relationship between the two. For example, there's been studies in China that have shown that there's a relationship between air pollution and COVID. Even simply a 10-unit increase, for example, of nitrogen dioxide had an impact on the rate of COVID infectivity. There's been other studies showing that there's an association between COVID fatality rates and air pollution. And I'm not going to go through all these studies. I know there's going to be another talk that's going to talk about some methodological challenges doing these studies and all the variables that you need to control for and other things. But, you know, there is a body of literature showing the association. In addition, the SARS outbreak and the MERS outbreak, also, there were some studies showing an association. So this association between air pollution and COVID is not specific to the COVID infection. It can also be shown that exposure to other types of infections, such as influenza, is associated with levels of air pollution. And then if we look at even rates of smoking, we know that that increases respiratory infections and impacts the development of COVID. So what I'd really like to spend a few minutes talking about is the why. And the common link is the immune system and the immune dysfunction that occurs when someone is exposed to air pollution and or COVID or the SARS virus. So one important thing to keep in mind, that when we're talking about inhaling, whether it's a virus, a bacteria, particle pollution, anything, any type of contaminant or dust, the size of the particle will determine how far down it goes into your respiratory tract and whether or not it actually reaches the base of your lungs and then crosses over into the bloodstream. So you can see that viruses are very small and will go, you know, to the depths of your respiratory tract. There we go. It wasn't working before. Whereas if you talk about particle pollution, PM10, for example, will only go partway down the respiratory tract. And that becomes important when we look at the alveolus at the base of the lung. And so this diagram shows someone inhaling wildfire smoke, most of which is PM 2.5. And those particles are small enough to go all the way down to the base of the lung and cause activation of different immune cells. One important immune cell is the macrophage, which in the blood is a monocyte. And the macrophage is one of our first lines of defense in addition to the dendritic cell. And it will try to engulf these particles and get rid of them. And this is a a picture of this in action. So label the circles, the dark gray circles are macrophages, and the black dots are particles of pollution, and the macrophages are trying to engulf the particles. And this activation of the macrophages causes kind of an inflammatory cascade throughout the body, and you get an increase in inflammatory markers and other proteins, and this results in widespread systemic inflammation. When we talk about the COVID-19, we know that the SARS virus attaches to an ACE2 receptor, and that this can occur at the alveolus level also. And again, you get a cascade of different immune cells reacting to the virus. And the SARS-CoV-2 in general sets up, again, a whole host of inflammatory changes within the body, causing a variety of increases in inflammatory biomarkers, changes in coagulation, hypercoagulability, and so on. And this generalized immune dysfunction results in a lot of the symptoms that you see with COVID. And so I'm just going to talk briefly about looking at the immune cells. There is a technology called CyTOF, which is a form of mass cytometry. It's a type of mass cytometry. And what's done is you look at someone's blood, you draw their blood, you isolate the PBMCs within the blood, and you use a technique to determine the types of immune cells and also the quantity of immune cells within someone's blood as single cell level. So once the PBMCs have been isolated, they're tagged with different metals, tagged for different antibodies. They are used to stain the cells, and then these cells that have been stained with the different metals are run through a mass cytometer. And at a single cell level, one cell at a time, it is determined which antibodies were present and which immune cells were present in a sample of blood. And what this allows us to do is to determine for a given sample the types of immune cells and the percentages. So in this immune cell plot, it's called a Wisny plot, we can see that the monocytes, which in the tissue are called the macrophages that we've been talking about, can be identified along with other types of immune cells, the CD4 and CD8 cells, B cells, and so on. And this becomes important because when we use it to look at the associations with pollution, and then there's studies being done obviously with COVID and using CyTOF, we're able to see at a very high level and with a lot of sensitivity what is going on in association with the environmental insults of either COVID or air pollution. And in general, what's being found is both of them result in similar immune inflammatory changes in immune dysfunction, accounting for some disease overlap with exposure to both air pollution and COVID. Thank you very much, and I'll stop sharing my screen.
Thank you very much, Mary. You kept your time very well, and you even made up a little bit of time. As we announced the questions and answers, we will save for the last part of the session today. So we will now directly go on to Tess Mursha. He's an associate professor at the Cincinnati Children's Hospital Medical Center and University of Cincinnati, where he leads the Population Genetics, Ancestry, and Bioinformatics Laboratory. His research combines quantitative ancestry and statistical genomics to unravel genetic and non-genetic contributions to complex diseases and racial disparities in human populations. And his particular interest is asthma and asthma-related allergic disorders. His long-term research goal is to understand how biologic predisposition and environmental exposures interact to shape racial disparities in health and disease. Some of his recent work includes the link between the COVID-19 pandemic, other health conditions, and chronic exposure to air pollution in the context of racial disparities. So Tess, we're glad you're here, and the floor is yours.
Thank you very much.
And please turn on your camera, Tess.
Yes, it is disabled. Can you?
Okay, that's a pity.
Yeah, it's disabled. I don't know.
Maybe Eva can undisable it.
Okay, so thank you, Aurora, for the nice introduction, and thank you for inviting me to talk to you all today. The camera is still disabled in any case, so okay, now I think it's working. So thank you again, Barbara, for the nice introduction, and thank you for inviting me for this really important talk meeting. So I think Mary presented very nicely the link between air pollution and COVID-19. I'm not going to go back to those things that I plan to discuss. The most important thing is that this small particles affect the immune cells, and the effects are not the same across board. So I'm going to talk to you about racial disparities. Although everybody has equal opportunity to be affected, there is disparities that we need to address in this pandemic. So I'll talk to that. So my talk is basically framed in a way that we, over the last just 1 year, we published multiple publications related to COVID. Actually, early on last April or May, we published in JACI about the link between air pollution, racial disparity, and COVID-19. And then we followed in different areas of interest. We looked at the pandemics from African continent perspective, and we saw much really the gaps or disparities compared to what we see here and over there. So we call it the African paradox. In any case, as you all know, COVID-19 is the third leading cause of death after heart disease and cancer in the US. So my talk will be around these articles, and if anyone missed my point, please refer back to these articles. So why is global prevalence difference? And if you look at this figure, I'm not sure you can see my cursor, but in any case, this is showing the global prevalence or number of cases across the different continents: Africa, the Americans, Asia, Europe, and Oceania. So if you look at I'm particularly interested to look at the African continent. If you look at the case for Africa, it's a really tiny piece on top of these massive epidemics. And also look at the death rate. The death rate is also a really tiny piece. If you look at the population, I will come back. It's actually over 1.23 billion population, but the death rate is not as you can see in other part of the world. So this number, I know this figure is a little a few months old, but I have seen the data just yesterday. There is some change, but the trend is more or less similar in any case. So in this talk, I will briefly outline why this global variation in COVID-19. I will give you some plausible hypotheses, including the climate hypothesis, the environmental neighborhood hypothesis, comorbidity hypothesis, the genetic hypothesis, and the evolutionary or natural selection hypothesis, and the immunity hypothesis, and the age hypothesis. These are really plausible hypotheses. Nobody knows for sure right now what causes this global variation in this pandemics, but we thought this is really important to explore further. So I call them this a plausible hypothesis. So if I start with the climate hypothesis, you can see that seasonality or climate could contribute to that. We have seen this in this sketch, in this figure. You can see the difference in different temperate zone. Mostly in North America, Europe, we have a high pandemics compared to the tropical region. In Africa, of course, there is also variation here in the eastern and western side. So you can think of the variation that is in South America and Brazil. But the interesting here is you can see there's a variation in pandemics. It's really in some cases related to altitude. We have seen publication that the higher altitudes, the lower the COVID epidemics. So we thought this has to do some. This could be a hypothesis that can be explored, whether seasonality, humidity, altitude could contribute. And as I mentioned, we have seen this in our own study in the publication we had in a global journal, Global Health. You can see this is African continent. There is an increase, but it's at a lower rate compared to the US, which is at increasing rate. Again, this is a little older figure, but the trend is more or less not that big difference. So as I mentioned, the African continent, over 1.3 billion people. And if you see the mortality rate, it's about 2.3 to 3%. So, the world population, about 70% of the world population live in Africa, but the total global case for COVID is about 3.6%. So I think the main point here I want to take back to the North America or Europe is that why is the variation even within Black community within US and Africa, and why is that? Genetically we expect to be more or less similar in terms of ancestral origin, but they have massive difference, in terms of COVID-19 pandemic. So the next hypothesis, the neighbor hypothesis, I think Mary mentioned that, how air pollution affect that, including PM2.5. So this figure actually was appeared a few years back in PNS. I think this makes really a perfect sense to look at back at this point, because if you look at the white population, what they cause, what they contribute to the environment in terms of PM2.5 is about, this higher percentage than what they are exposed to. That means they exposed a lot of PM2.5 to the environment and to other community rather than to themselves. They live in a better environment. Whereas if you look at Hispanic, they are exposed actually much higher to the PM2.5 compared to what they contribute to the environment. The same is true for Black. They are exposed over 56% than they cause to the environment. Whereas the white, as I mentioned, they are exposed less than 70% than they exposed. So that means in the neighborhood, the Blacks or the Hispanics, they live actually is in a much worse environment rather than which is contributed probably by the other white and other population. So this is really a very good data to see what can we make adjustment in order to change this paradigm. So we also look at in our own study, we look at the state of Illinois. We look group Chicago area by zip code and county. As you can see here, the clustering is actually not by county, rather by zip code. So we have a really granularity of a very zoomed area where COVID is at high risk or than at other areas. For example, we have these two clusters. Cluster one, higher confirmed case of COVID, higher Black population, high food stamp, which is a perfect environment for COVID. Whereas cluster two is this one, on the left side, which is lower confirmed case, higher white population, and lower blacks, and lower food stamp. So actually, we cluster much at the zip code level, gives us a more granular data than a county level, because this county probably fall in the same area than other areas of this map. So that's why some years back, actually, whether there's an article of USA Today which says whether your zip code is important than your neighborhood or gene code. So the point here is targeting high risk neighborhood is really important than is not just high risk individual. We need to target the neighborhood, not just the genetics or the individual environment or the individual per individuals. So neighborhood is really important to be addressed in this case. So the comorbidity hypothesis is another hypothesis we draw in, and this is as Mary and other will talk about these details. I'm not going to the details, but the comorbidity hypothesis is being a diabetes, obesity, and all other cardiovascular risks, including respiratory disease, is a risk factor for COVID-19. So I also mentioned race ethnicity, which is also has something to do with racial disparities, is another link that linked to social determinants, basically feedback to other risk factors. So this is a complicated chart, but it tells us how difficult to unravel each of these, but eventually we need to address those factors if you have to have a really address racial disparities that exist in the country. So there's also another talk toward the end, which about genetic hypothesis. Some mentioned at the beginning of the pandemic, racial disparity could be as a result of the receptor SEA2 gene polymorphism, and this is because we know that SEA2 gene is in a receptor that the entry point for the SARS-CoV-2 to the human body. So this is an important receptor gene, and at the beginning we all thought that this is probably the most important factor that leads racial disparities in human. But as most study showed in the past, I'm sure there's a lot of studies. This is a little older document from a publication from the England Journal of Medicine. So actually, that SC2 gene is not actually in this Manhattan plot. So there is other factors we need to consider other than a single receptor gene. So I would like to mention that this is a small sample size compared to what US entail at this point. So this is just a preliminary study, but it shows you that the low-hanging fruit expected, like the SEA2 gene, is not actually in this candidate list. So another important point is the evolutionary and natural selection hypothesis, the Th2 to Th1 cytokine response due to infection. So as we all know, living in the tropics, in the temperate area, where low exposure to worms and other pathogens, and basically we are protected from many external factors, including we have the hygiene hypothesis that we all think is an important for that case. So if you live in a more tropical area, like in Africa, you have a high exposure to worms and other pathogens. That helps, that serves as a training for the new pandemics that's coming to exist. So that's part of the equation, that the cytokine storm is occurring in a Western society compared to Africa, which leads a much massive difference in terms of risk factor. So the pre-existing immunity probably in Africa could contribute the low pandemic so far in Africa. So this has to do with evolutionary adaptation. So the final, the other hypothesis is trained immunity hypothesis, the BCG. As you know, this is vaccination that is common in most developing countries to malaria and to protect malaria infection, and this has been going in many countries. So I was looking at the map of vaccinating region in the world versus the COVID-19. I don't think we can make a one-to-one association here, but there is some similarity that the higher the vaccine coverage, the lower the COVID-19 epidemics. This has to do with trained immunity, as I already mentioned before, but it needs a much more study in order to really make a more clearer association of this kind of relationship. Finally, as you all know, the age hypothesis is another important factor, that we all know that being 75 older is the highest risk group as far as COVID-19 is concerned compared to younger age groups. So why is that different? I know there is also sex difference. I'm not going to that at this point, but as you can see here, why age different? This makes a big gap in terms of COVID-19. There are a number of issues, a number of factors. One could be presence of comorbidities in adult. Adults usually have more type 2 diabetes, other cardiovascular, obesity. These are comorbid population, and comorbidities one factor is an important factor of COVID-19 compared to young children. And also, as I mentioned, the SCE2 gene increase, the expression of SCE2 gene increase with age, and that could be also another factor that could contribute in the immune system. As I mentioned here, the immune system is deteriorated as we age. So what are the opportunities and the challenges to address? I think there are multiple levels we need to address. As I mentioned here, individual level is important. We all are different in terms of genetic code. There's some share of individual differences, and neighborhood factors. Neighborhood is not the fault of individual. We need to have a very good neighborhood in terms of exposure and safety, all other factors which has a health impact. And of course, as a national level, we need to have a strategy to mitigate and address racial disparities that's going on for long. And these are really, some of the things are controllable and some are not, but we need to address as much as we can. In summary, I think COVID-19 disparities is high in minorities, as I mentioned, and this reinforces actually the known health disparities that in the community. I study asthma, and asthma, there's a 4 or 5-fold difference between white and black, but nobody pay attention. Probably it's under the scientific community. We all understand that, but COVID-19 right now is actually exposed to the society level, to the to a more and a more understandable way at the community level, at the society level. So this is an important time. I believe it's an important time to address health disparity in all form and in its all forms. So underprivileged or minority population are more likely because they live in an area with high exposure to air pollution. As I mentioned, they live near highways and industries. They are obese or suffer from comorbidities, including diabetes, cardiovascular, and they are life-threatening. The high to get hospitalized and get life-threatening COVID is high in those communities. So the solution, as I mentioned, is it should be a multi-level intervention, and there's no one thing fit for everything. You need to have a multi-level intervention, and I believe health disparities are preventable or at least modifiable at this time. So thank you very much. I'll be happy to take questions after the final talk. Thank you.
Well, thank you very much, Tess. You've given us a lot to think about in your talk. That was very interesting. I'd like to switch over now to Paul Villeneuve, who will talk about methodological challenges of doing COVID-19 research in relation to air pollution. Paul is a professor in the School of Mathematics and Statistics at Carleton University and holds an adjunct appointment in the Department of Public Health Sciences at Queen's University. Before that, he was senior research scientist at Health Canada. His research is focused on occupational and environmental epidemiology, and his current program includes research on health impacts of air pollution, greenness, and urban built environments, occupational exposures to diesel, and radiation health effects. Dr. Villeneuve received his PhD in epidemiology from the University of Toronto, and he is currently on the editorial boards of Environmental Research and the Canadian Journal of Public Health. And I hope we could solve all the technical issues we had before this session, and that you can start your presentation now. There it is, Paul. Yeah, the floor is yours.
That's good. Thank you very much. And thank you for the invitation to present today with the final lineup of speakers. So today I'm going to be talking about issues more from an epidemiological perspective in terms of studying relationships between air pollution and COVID. And I'm going to be drawing primarily on examples from Canada and the United States, though I think a lot of the issues that I raise are relevant to other regions as well too. So just to backtrack for a little bit, and I think it's only fitting in a way that the first cases of COVID in Canada really emanated from a curling tournament out in Alberta, and also in the maritime regions, which has handled things really well in terms of public health interventions from funerals. And so the first outbreak out in Newfoundland was really driven by some interactions at this funeral. Now, when we think at the onset of the epidemic in terms of what might some of the risk factors be, and I think at that point it was pretty well established that it involves person-to-person contact. Public health measures can play an important role. Travel, some occupation. And, the thinking, I don't think at the very beginning in terms of doing contact tracing on people at this bonspiel, the curling tournament or the funerals was asking people about their air pollution levels and their air pollution levels over, their last 20 years or what have you. But some of the evidence that has come out, certainly showed that people with advanced comorbid condition are more susceptible to adverse outcomes from COVID. So I do think there is a role for a mediating effect on air pollution for more advanced forms. And Dr. Pernicki gave a really nice overview of that and some of the potential direct mechanisms whereby air pollution can affect COVID. Now, one of the first studies that came out was an ecological study. And I think that relative to some of the other correlational studies that came and were published on the SARS and elsewhere around the world, a much stronger design and approach to analysis. And they reported that there's an increase of 15% of COVID mortality per in 1 unit increase of PM2.5. And then after adjusting at a county level for some of their risk factors, that risk came down. And there was a similar study that came out of Canada that found that air pollution increased COVID incidence. And, this prompted, some thoughtful consideration on the part of myself and Dr. Mark Goldberg. And we published an editorial about some of the methodological challenges of looking at this question. And what struck us at that point was that there's really a poor ascertainment of COVID incidence and mortality, particularly at the early part of the pandemic. You know, the messaging that a lot of the public health units in Canada were given was that we're really missing 20 to, we're under ascertaining by a factor of 10 to 20 fold the number of cases. There was estimates that in April last year, we had missed 100,000 incident cases of COVID in Canada. And even now, death certificates in Canada reflect those who are tested for COVID. So, you know, we have situations in Ontario where things are being poorly managed, where people are dying at homes, and that's not, they're not getting diagnosed. And we have an instance, for example, of a 13-year-old earlier this week who died at home because her family didn't want to transfer to a hospital across the province. So there's a lot of issues in terms of ascertainment, and this impacts hospitalization data, incidence data, mortality data. There's also issues around correlated data. And a number of studies have shown the impacts of super spreader events. So, you know, another consideration about some of these ecological factors, and this was really nicely described by Dr. Mercer as well, too, is that there's a lot of factors that in terms of inequities, in terms of racial variabilities, that we can't really control for well with some of these ecological studies. Now, one of the things that we raised in our editorial group was this issue of timing on a pandemic curve. And at the early onset of the pandemic, this could really affect comparisons when we look at air pollution and COVID, because the larger cities tended to be the first areas that are impacted. And if we were to look at some of the reports from the CDC that you see in this figure, that shows in terms of the pandemic curve, you see a greater proportion of cases occurring in urban areas at the beginning, but they were overtaken by rates in more rural areas. And I think what's also important is that there's a lot of variations within these regions in terms of the public health measures that are implemented. There's transmissible variants that are coming out. So this yields a lot of complexities for looking at these associations, especially with ecological studies. And I think one of the key takeaway messages is that the findings from these ecological studies will vary over time. And while there have been some attempts to control for peaks on this pandemic curve, you'll see, for example, this curve, the red curve for Ontario, that these curves are multimodal. And relative to areas that have gone for a COVID zero approach, such as Australia, you can see how important some of those public health measures are to controlling the pandemic. So we recently updated some of the analysis that was published for the US county. And again, the group down in Boston did a wonderful job in terms of documenting some of the data sources down there. But when we updated the data to include cases through March 2021, you'll see that the shape of the curve changed. And we actually find that there's a nonlinear relationship between COVID and air pollution. And in fact, for most counties, as you go up, there's reduced COVID with increasing PM 2.5. So again, this illustrates the importance of the length of follow-up time. But it also, I think, highlights a concern about some of the ecological studies. And, you know, as an illustration, we also did analysis of prevalent HIV and ambient PM 2.5. So again, ecological data, in this case, our analysis using these county level data of PM and adjusting for some of the same county level covariates seems to suggest that PM 2.5 reduces the risk of HIV. So I think there needs to be caution in terms of some of the messages that come out of these ecological studies. So more recently, some have moved to more highly spatially resolved interurban comparisons. And a colleague from Health Canada, Dave Steeb, has just published a paper showing that an increase in reactive oxygen species PM 2.5 is associated with an increase in PM incidence. And there are a lot of advantages, I think, to doing interurban comparisons. The public health measures are more standardized in a smaller area. You can have possible access to individual level data, higher spatial resolution. of air pollution. So there's a lot of advantages to moving to interurban studies. But I still think there should be some caution given to these studies. And I think source of transmission matters. So as an example, let's take Barrie, Ontario, that had an outbreak in a long-term care facility, elderly individuals in the winter, January, February, 70 deaths, over 200 infected. Barrie's a relatively small city, but it accounted for most of the COVID cases in Barrie at this time. And the outbreak was due to person-to-person contact in a long-term home. So if we were to look at the NO2 exposure at that home, it's 13.3 parts per billion based on an exposure in 2016. All other postal codes in Barrie had an average exposure of 8.8. So if we fail to consider the source of the exposure, we may falsely begin to attribute air pollution risks to these outbreaks. Now, there's been a lot of other outbreaks across the country, as there has been worldwide. You know, one of the biggest in Canada was that at the Cargill meat packers in Alberta. Over 1,500 cases, accounting for 20% of Alberta's total in a meatpacking plant. So these occupational exposures are important drivers. There are connections between regions. Newfoundland, Alberta, for example, a lot of workers in the Alberta oil sands come from Newfoundland. So there's been spread in Newfoundland, but it's been due to some of that occupational exposure. Something else to keep in mind about outbreaks. They can really drive COVID cases in a city. And in Toronto, for example, there's been a number of outbreaks at Amazon factory, multiple outbreaks in meatpacking plants, Canada Post indoor working environments. And even today, they released a newspaper article that showed there's outbreaks in 20 homeless shelters. So a lot of these cases and incidents is really driven by occupational spread. And a lot of, in Toronto, a lot of workers in these places tend to be minority populations living in intergenerational homes, and they live in areas of high air pollution. So it becomes really important to try to understand what's going on in terms of these other risk factors. And this map, for example, will show you attempts to vaccinate in Toronto with pharmacy programs. And you'll see these red dots are all pharmacies that are offering new vaccine programs, but guess what? They're all outside of these lower SES areas that have high air pollution levels and high COVID rates. So there's a number of different inequities in terms of COVID in Ontario. This just kind of highlights some of those. And right now that the government is struggling to implement policies that protect workers. So I'm going to
3 minutes.
Okay.
You have
So
3 minutes.
I'm going to scroll down a little bit. And what I also want to do here is highlight some of the important influences of vaccines in terms of altering changes in COVID rates. And these can play an important role as well too. And it becomes important to try to account for these in our epidemiological studies, as well as changing distributions of variants over time. So there's been a few studies that have highlighted that the outdoor transmission of air pollution is much lower than transmissions in indoor environments, and that only a small proportion of the cases through contact tracing are actually occurring through outdoor transmissions. And if I were to think of some of the studies, features of data we'd like to include in these studies of air pollution, individual level data is really helpful. Information on the type of variants, the vaccines, as well as statistical methods for correlated data. So it leads me to think that if we were to try to look at this question, the case control study may represent the most viable way for doing so. And I'd just like to sort of mention a few things about the policy implications. And our governments have struggled with implementing policies to protect residents. And one of the things that they did in Ontario was shut down outdoor activities about a week and a half ago. And this might not be the wisest thing, given that indoor environments may be an important source of spread. And the last thought I'd say about many of these studies is that sometimes the types of data we have don't allow us to answer the question. And I think we need to be cognizant of some of the limitations and put the air pollution findings in their context with some of the other risk factors. And a more detailed description of some of these issues has been described in our paper. Thank you.
Thank you very much, Paul, for keeping the time and getting these messages through in this short time span. Yeah, we are now switching gear again and taking a broader, more global perspective, and we will do so by moving on to the Netherlands and looking at an example where animal health and human health is being connected. And this will be done by Dr. Lidwien Smit. She's an environmental epidemiologist and associate professor at the Institute for Risk Assessment Sciences of the Utrecht University in the Netherlands. Her research and teaching focuses on environmental and occupational risk factors for human health in the context of One Health by studying public health threats at the human-animal environment interface. She's a principal investigator in a long-running study on agricultural air pollution, respiratory health, and zoonotic infections. During the COVID pandemic, she has led unique studies on environmental exposures to SARS-CoV-2 in mink farms and meat processing plants. Lidwien, we're happy to have you with us, and the floor is yours.
Thank you very much, Barbara, for the introduction. And also thank you for the invitation to speak here today. So in my presentation, I will discuss COVID-19 from a One Health perspective, meaning that to understand the pandemic, we must not only study humans, but also wild and domestic animals, and also our broader environmental context. And so in this picture, you can see a Chinese market where live animals are sold. And those are seen as a hotspot for the transmission of emerging pathogens from animals to humans. But I will also take you on a journey to European livestock farms where SARS-CoV-2 infections were found in minks. So One Health is the term that is used to acknowledge that human, animal, and environmental health are tightly interconnected and that they need to be studied in the context of each other. One example is antimicrobial resistance, because antibiotics are used both in animals and humans, often the same types of antimicrobials, and with overuse of antimicrobials can cause antimicrobial resistance, and it can be both for veterinary and human medicine a problem in treating infections. And those antimicrobial resistant bacteria, they can spread either through the food chain by eating food that is contaminated, or through the environment, even through the air. Another thing is that many pathogens that affect humans have emerged or re-emerged over the last decades. And so both the total number and the diversity of infectious disease outbreaks, they increased significantly since the 1980s. More than 60% of those emerging infectious diseases are zoonotic infections. So they originate from animals and animal reservoirs. One of those animals that are often mentioned are bats. And well, bats are known to harbor many zoonotic viruses. And they're the natural host of both SARS and MERS, but also other viruses like rabies or Ebola. And well, they're the only flying mammals, but they have several aspects in their ecology that also favors virus circulation. So worldwide, there are large bird populations, and they're one of the most numerous mammals around the world, and they live in very dense communities. But most coronaviruses don't transmit directly from bats to humans. They need other intermediate hosts, and for example, civet cats have transmitted SARS to humans, and dromedary camels can transmit the MERS virus to humans. For SARS-2, however, the intermediate host remains to be elucidated. So this is the taxonomy of coronaviruses. It's a large family of coronaviruses. You have alpha, beta, gamma, and delta coronavirus genera. Some are only found in animals, like in birds and pigs, if you look at the gamma and delta clades. But there are 7 coronaviruses that are found in humans, and 4 of them only cause common colds. But 3 of them, MERS and SARS and SARS-CoV-2, are able to cause very serious respiratory disease. So at the end of March this year, the WHO reported a very interesting report. It was a report of a search for the origins of SARS-CoV-2 in China, which was done by a One Health approach, because many experts from very different disciplines were looking for the origins by looking at available data and all kinds of putting data together. And one of the figures in this report, which is really a very interesting read, is this conceptual framework of the possible routes of SARS-CoV-2 emergence. And introduction through direct zoonotic spillover was one of the options that was looked at, and it was seen as possible to likely. So that would mean that you have a bat, and the bat would transmit, or another animal would transmit directly to the humans, and then humans would transmit it to each other, possibly through super spreader events, like at a market in Huanan. But a more likely option is that an intermediate host was at play. So a bat or another animal would first transmit the virus to another animal that would get in contact with a human, either directly or through the food chain. And then the humans would be able to transmit it among each other, possibly after evolutionary adaptation. And that was seen as the likeliest, or the most, the very likely route of introduction. Although until now, no evidence has been found in a lot of animals were studied, like serology was done and all kinds of tests. But there has not been any evidence on the origin or the animal that has originally transmitted SARS-CoV-2 to humans. The food chain was also looked at as an option, and in that case, also first there would have been transmission to animals, then to the food chain, and then to humans. But this has been seen as a less likely option. It's possible, but respiratory transmission is much more likely. Finally, laboratory incident was investigated as one of the introductory pathways, but that was seen as extremely unlikely because at none of the laboratories any virus was studied that was even close to SARS-CoV-2 before December 2019. Well, of course, it's important and interesting to know the origin of SARS-CoV-2, but we should also think why we should study COVID-19 further in animals, so using a One Health approach. Well, one of those reasons is that, of course, we want to prevent if an infectious disease like COVID-19 emerges in humans, that there is further transmission between animals or animals to humans or back from humans to animals. I mean, think of exposure to pets or domestic livestock animals, or also to wildlife. Another reason is that we need to study and monitor new mutations when interspecies transmissions occur, because the virus may adapt to new species and they may cause dangerous new mutations. And another reason to study this is to prevent establishment of new zoonotic reservoirs. I mean, if the reproduction rate is going down in humans by vaccination or by preventive measures, and you keep on having zoonotic reservoirs from which you can have spill back to the human population, it's much more difficult to contain an infectious disease. And finally, also, we need to study this kind of diseases in animals to prevent future spillovers and to learn from those infectious disease epidemiology. So if you look at the timeline of the first reports of COVID-19 in animals in the early months of 2020, the first report was in a dog in Hong Kong, and then also early March, several animals were studied by the Hong Kong authorities. They found a dog and a cat to be infected with COVID-19. In March, a diseased cat in Belgium was found to have COVID-19. And then also experimental studies were published showing that cats and ferrets, they can not only be infected, but they can also transmit the infection between each other. And well, dogs can also be infected, but it's more difficult to transmit it between each other. This study also, or another study also showed a rather high percentage of seropositive cats in Wuhan. Then in April, zoo animals in Bronx Zoo were found to be infected with COVID-19. So tigers and lions, they probably had got the virus from their caretakers. And then end of April, almost a year ago, or more than a year ago, the first infected mink farm in the Netherlands was found. So this was the first commercial farm where animals were infected. There were soon after that also experimental studies published showing that bats, certain fruit bats, but hamsters and rabbits can be infected. But fortunately, pigs, chickens, and ducks are not susceptible to be infected with this virus. And by the end of June, already 17 mink farms in the Netherlands were infected. And also in Denmark, 2 infected mink farms were found. So we will now zoom in to the Netherlands. It's a country where I am from, and it's a very interesting place to live if you want to study One Health. It's a very densely populated country, 17 million people on a rather small surface. And we have our share of both urban and agricultural air pollution. This is more urban area in the west of the country. And here in the east and in the southeast, especially, are many livestock farms. So this map shows reactive nitrogen deposition. And well, from traffic, it's mainly nitrogen oxides, but from farming, it's mainly ammonia pollution. And this is in the southeast, also the area where there are many livestock farms and where the first mink farms were detected to have SARS-CoV-2. But it's also an area with a history of other animal diseases like classical swine fever, foot and mouth disease, and some zoonotic transmission events from avian influenza, for example, or Q fever.
Litwin, you have 3 minutes left.
So COVID-19 in the Netherlands showed very remarkable spatial correlations. Hospitalizations were mainly seen in the southeast in the first month of the epidemic. And people already started to notice the correlation with farms like pig farms, but also ammonia air pollution, and also with earlier epidemics of zoonotic diseases. However, there were also other covariant factors that were very important. Like in the Southeast, school vacations were in the Southeast were the last. And many people from the Southeast had traveled to Italy or to Austria, where they had picked up COVID-19 in the early phase of the epidemic. Also, the Carnival is a festival that took place in the same time and also mainly in the southeast of the country, and it really worked like a super spreader event. So until now, it's really very limited data from the Netherlands that have been analyzed in a proper way, because those kind of factors cannot be taken into account, and publications showing that air pollution is associated are mainly ecological. Well, as I said, in the same southeastern area, there were a lot of concerns regarding this high incidence of COVID-19, and it co-occurred with SARS-CoV-2 in mink farms. And because of the concerns, the government immediately decided to set up a One Health consortium to study, both from animal, environmental, and human perspective, these outbreaks in mink farms. This is a mink with pneumonia from COVID-19. Mink farms were detected because of increased mortality. And it started only with a few mink farms, but it spread very easily through the southeast of the country, despite preventive measures. And mink farming is now banned in the Netherlands. Well, we also found mink to cat transmission and also mink to dog transmission on those farms, because those animals roam freely in those contaminated mink farms. And we took several environmental samples of SARS-CoV-2, both inside the mink farms, but we also did air sampling, so indoor air sampling, both personal and stationary, where we found contaminated air inside the farms. But outside the farm, so in the outer air, there was not no SARS-CoV-2 RNA detected. In humans, 68% of the mink farm workers at the first 16 farms were infected with SARS-2, and they were clearly found to be associated with mink, looking at the whole genome sequences, but also at epidemiological information. We found the first evidence of direct animal to human infection. And that was only found in mink farmers, but not in the human population living nearby. So there was no evidence of secondary transmission or environmental contamination that had led to humans nearby being affected.
Litwin, last minute, okay.
Yeah, this is my final slide, thank you.
Perfect.
So what are the lessons that we need to study for future challenges like COVID-19 or other future emerging infections? Well, I think One Health is very similar as planetary health. It's a very similar concept of looking at environmental and human and animal health problems. If you look at from the human side, there is enormous population growth, which makes us much more vulnerable for infections, but also traveling to natural areas where there is wildlife from which we can get infections, increasing urbanizations, and also our increasing food demand from animal sources is definitely leading to issues that we must study from a One Health point of view. So to conclude, it's really imperative that animal, public health, and environmental scientists and also authorities, governments work together to better understand, but also to prevent emerging infectious diseases. But also non-infectious diseases could be very well studied using this framework. So thank you very much.
Thank you very much, Litwin, and for giving us this perspective on a One Health or planetary health approach. I would now like to hand over to Jeff Brook, who is going to moderate the Q&A session. Jeff, please.
Hi, Barbara. Yes, thank you very much. And thanks a lot to all the speakers. This was a really informative session, a challenging topic, and I'd say relative to the other conference, I haven't seen as many questions streaming in, but still plenty of really good ones. And so I'll sort of jump right into one of the first questions that came in relation to Tessa's presentation, which was around what we're observing now in India. And there was sort of a slower increase there, but now it's raging, and whether or not Africa is just delayed in what's going to eventually happen there.
Yes, I think that might be the case. The delayed situation in Africa is actually now it's a little bit surging in some countries in Africa as well, but the India case is really troublesome because we are getting more vaccine, but things getting out of control right now, as I heard this morning from NPR. So I think it's a very good early call for most African countries to take it this very seriously. They are not out of this pandemic because of what happened last year may not be the same as moving forward. So yeah, I believe given the the geographic similarity and the population living standard is somehow similar, I believe Africa will need to take action, especially social distancing, all those public health measures, because vaccinating 1.3 billion will not happen in any time soon. So the best measure is to take action in terms of public health measures, the simple case that can protect over 90% of the pandemic. Yeah, thank you.
Yeah, right. Thank you very much. Question for Mary. Mary, was great to hear more details of immune system response. Do you have a sense as to how much air pollution exposure might be enough to alter immune system response, or the timing of the exposure, whether or not inhaling air pollution at the same time as being exposed to virus or before is having a different impact, and whether or not maybe different sources like people are spending a lot of time indoors now. Maybe cooking fumes are particles that may ramp up the immune response and make you susceptible.
Right. Yeah, the cooking fumes is interesting. The natural, using natural gas, there's interest in looking at other toxins that come off, like formaldehyde, that hasn't really been studied, and how it alters our immune system when we're exposed to a natural gas stove. There's obviously things we don't know. What we do know, we actually just published a paper looking at 6 to 8-year-old children, and we found that certain immune cells actually were associated with pollution even 1 day after exposure. So it depends on the immune cell type, and it depends on which pollutant, but certainly there are changes that can be seen quickly. And, this makes sense in terms of when someone's exposed to wildfire smoke, the majority of the outcomes occur within that 2 to 3 day period of being exposed. So obviously, the health impacts can be appreciated fairly quickly. In terms of which pollutants and which ones might be more toxic, I think that's, there's been studies in air pollution. Most of it the majority has been focused on PM 2.5, but there's other awfully ozone and other pollutants that we know somewhat about. There's been studies showing that things like high blood pressure in an adult, you can see that association with air pollution, and you're under the criteria guidelines that the EPA has set. So, most likely the levels of pollutant criteria are possibly too high, since we can see associations even below those levels. But, I think overall there's still work to be done in the area to know exactly which pollutants and which time frames are most critical.
Yeah, it seems certainly with tools you have now to look at the immune system response, there's a lot lot of potential there to follow up more. Thank you. Paul, I think your quote from John Tukey at the end struck a chord. It certainly did with me. And one of the questions that came, was that your point was very well taken about John Tukey's quote, helping to sum up the aggregate weakness of our collective COVID-19 learning. Positivity rate is problematic, as you pointed out, but useful. And it often is the only available metric for public health reasoning. But overall, do we need more money and efforts focused on study populations with comprehensive RC-PCR test screening of people to achieve this and get more look at the asymptomatic and symptomatic populations, to advance learning here? Or are there other ways?
I guess, I guess one of the questions I'd have is, whether that question pertains to sort of understanding the role of outdoor air pollution and the spread of that. And I guess my concern is that there are so many challenges with the different variants, the different vaccine uptakes, the different social determinants of health, that it becomes a very tough question to answer. So I think John Tukey's quote holds. I do think it's really important to get a better sense in terms of prevalence, asymptomatic cases, particularly as in Canada, we're thinking of going back to schools as well too, and figuring out ways to sort of do mass screening, and all of that being done at the same time when vaccines are rolling out in different priority populations. It's really tough to disentangle all of those factors.
Yeah, and I wonder, as you talk about the temporal changes that have been happening throughout the pandemic, in so many different aspects, introducing vaccines is one of the latest ones, and how it's introduced. So the data only seem to be even more complicated, in future retrospective analyses. Do you think there's a path forward, retrospectively, or is it just going to remain so complicated?
I haven't figured out, how best to analyze it. One of the slides I skimmed through quickly, because I know I was on time, was the issue of the short-term studies of air pollution and sort of the challenges there. And, one of my slides displayed how much day-to-day testing in Ottawa varies in terms of sheer numbers, but also in terms of the criteria. At the beginning of the pandemic, you couldn't get tested unless you had direct contact with a case, or if you were traveling from abroad. And now if you look at the guidance on Ottawa Public Health website today, there's like about 20 different criteria. So trying to disentangle the day-to-day changes in a time series or case crossover study becomes very challenging. So, I think, yes, potentially maybe with a case control study, we might be able to answer some of those questions, but I think we also need to look at things like contact tracing. And if contact tracing data tells us that none of these spreads are happening in outdoor environments, we need to begin to take that at face value and sort of learn from that evidence as well.
All right. Thank you very much. A question for Dr. Smit. In terms of sharing information that's been seen around mink farms, and we heard about that, I think, in Denmark and other Scandinavian countries early on also. And this is clearly high density of animals and people together. What so far is being learned about that, and any changes that have been made around the mink farms that might help with the next pandemic?
Regarding the mink farms, in the Netherlands, the government has at some point decided to ban mink farming completely. Mink farming was already going to be banned as of 2024, and it's now, been earlier. It's in 2021. It was banned for good in the Netherlands, but in other countries, other choices are being made. They simply hope that after containing the pandemic, they can start mink farming again. So I'm not sure whether a lot of lessons are learned. Also, other farmers in the Netherlands, they often point to like Asian livestock markets, and they fail to see in a way that it's really the same kind of issues that can occur despite the better biosecurity in those intensive livestock farms.
Yeah, very challenging. So a question that's been in my mind, and Mary alluded to it a little bit, and maybe all the panelists will have some insight. It's a difficult one, probably more an aerosol scientist question, is around air pollution's role being a vessel for the viruses to attach to and then be inhaled and potentially infect a person. What's known about that potential mechanism as maybe explaining associations that may be there? Does anyone have any insight on that?
I can give a short comment on it. I know there was a paper that came out, I believe, by SETI saying that they found the virus attached to the particle pollution. And we do know for particulate matter, the composition of it depends on what's being, what's it's being formed from. And we know things like heavy metals can attach to particulate matter and kind of hitchhike into the body that way. And so I would, I don't know the full science on the attachment of the virus. It seems like it would be possible. And no, I lost my train of thought. I'll stop there.
Any other panelists have a thought on that one? There's so much coming out so quickly in this field that you hear things and you forget where you hear them, and you don't know whether they're published or just an early report. Yes.
Yeah, I can just add a little bit of what Mary said. I think most of the study so far, and it's all based on association, I would say. There's no causality so far. It's just too early to say whether the pollution has something to do with COVID. One thing I am for sure in terms of racial disparity, that polluted environments are mostly inhabited by minorities, and then pollution becomes, a surrogate for other factors, like, all factors that related to living standard or, income, socioeconomic standard, racism, all other factors could be contribute through the environment you live. But I personally don't think pollution directly has an impact. There may be, but that needs some causality using a mouse model. In our case, we do a lot of immunology work using a mouse model. But the problem is we do one molecule at a time. So we can't really have all pollutants at a time. We do PM2.5 and then we leave the rest aside. So I don't know how much that contributes, but at least we can establish some causality from that perspective.
Thanks very much. One last question. And this has to do with we've observed in many areas with lockdowns, improvements in air quality that may or may not be leading to improvements in health. Is this an area where there's some potential, this sort of great natural experiment that's happening to learn more about air pollution and health? Is there a path forward for that? Maybe Paul, epidemiologically, you could think about that some more, but open to anyone in the panel.
Yeah, no, and I think that's a good point. And I think there are surely some sort of benefits. Again, the challenges epidemiologically in studying that there are many. People may be avoiding going to the hospital because they don't want to catch COVID. Not only is air pollution being lowered, but the way that COVID is operating is affecting many of our behaviors and our normal inclination to go get healthcare. So things may get delayed. You think of like cancer screening, like there's a whole wide variety where lockdowns have impacted our health. So trying to, again, separate out the reduction in air pollution from those other behaviors that are going on becomes challenging.
And any follow-up from the other panelists?
Well, I'd like to add it. I agree. COVID makes it extremely challenging given the lockdowns. there have been studies prior, some studies, I believe it was the Beijing Olympics, where they shut the city down. And by doing that, the pollution decreased, and they were able to see reductions in ER visits and hospitalizations for asthma almost immediately. So I think that the overlay of COVID makes it really messy to try to pull out those associations. But maybe if we look to former situations where there's been a different reason for a reduction in pollution and we've seen improvements in health, might shed some light.
Right. Yeah, this question of accountability is an important one for HEI and many stakeholders to look at, but this might be just too difficult of an experiment. Time will tell. Just to sort of wrap up, the question of whether air pollution influences COVID infection or outcomes is really a difficult one. And Paul provided some reasons to be cautious in interpreting the early epidemiological studies. Beyond this, a challenging question to wrestle with is, how decision makers should respond to such evidence as the virus continues to infect people and really have serious effects. The challenges of the coronavirus brings reveals are numerous, going way beyond air pollution. As Tess Mersha, his presentation gave clear examples of the extent of racial disparities and the importance of neighborhood and social determinants of health. And there are likely new insights that can be gained about air pollution health in regards to the immune system that Mary showed were very interesting in those areas. And they are sort of backstopped by earlier work on air pollution and other respiratory infections like pneumonia. But we know that the lessons from COVID-19 are much broader. That the human impacts of the environment are very broad, and that reminds us of the importance of the One Health view that Ludwin presented in that area. I think a critical thing to keep in mind. But another key lesson that we're learning from COVID is how quickly the world can actually change in a crisis. And many parallels to this global response are being suggested regarding other comparable challenges, from the climate crisis to homelessness. Clean air has been well documented. We talked about whether or not we might be able to see the benefits of that. Certainly very difficult. But while it's important to understand and address air pollution health effects, in the grand scheme of things, and many of us, I think, through this time are thinking a lot about the grand scheme of things. A question that's really important to ask is, what is changing? And clearly the world has changed. Can't get around that at all. Just consider what we're doing today. We're successfully engaging in a second HEI annual conference with a smaller environmental footprint. The whole experience is not possible online, of course, but we are learning and adapting. And I'm going to turn it over now to Paul Dan to sort of wrap up this session. Thank you very much to the speakers again for covering this topic in such breadth. And thanks, Barbara, for taking us to this point.
Thanks, Jeff. And I just want to thank both the chairs and thank all the speakers for their presentations, and thank everyone for attending. As I said at the beginning and on this slide here, the presentations and the recordings will be on our website in the next few weeks. Our next webinar is going to be on May 4th. That's going to be on From Evidence to Action, Synthesizing Air Quality Evidence Relevant to Public Health. And again, please do fill out the post-webinar survey and let us know what worked and what didn't work. Thanks a lot.
Thanks again, everybody. Have a good rest of the day or evening.